I'm Liz. I am from Cameroon, and here at Cornell, I'm a fourth year PhD candidate in biomedical engineering. What I do for my research is that I study how you could use your DNA or RNA or any other nucleic acids to look into how your whole system is doing, meaning your organs, going from your kidney to even your lungs. And we're also looking into infections, and all of that is going through the motions and looking at particular things in your body, such as your proteins. Or when you talk about proteins, you talk also about enzymes, because enzymes are entities that are very important for your whole system and for us also as researchers, because without the enzymes, we cannot digest the proteins that we want to use and we cannot get to the DNA that we use in our diagnosis. And here today, we are going to talk about enzymes, and we are going to be making a little activity for which you could look into the lesson plan that has already been prepared for you to work through. And since we are talking about enzyme on that subject, let me connect it to something that you know. Let's say you're going to the market, you bought some bread and you're eating it. While you're eating the bread, you might realize that the bread is a little sweet. Why is the bread sweet? If you eat it fast, you won't realize that. But if you take your time, you will realize that your bread is becoming sweeter the more it stays in your mouth. That is because in your saliva, there is an enzyme that breaks down the sugars, and that's why you're able to taste it. And same thing with when you're digesting your food. The only reason why you're getting the nutrients that you need from the food is because you have enzymes in your body, in your stomach, in your intestine, and in your mouth that break down the food into different components that your body is able to use and digest. And while we are on the subject, before we start the activity, let me tell you one thing is that safety comes first when you're doing experiments. So you have to make sure that you're always wearing your gloves. You have a nice protective gear, like a lab coat, for example, and you're protecting your skin, you're protecting your hands, you're protecting your eyes. I have my eyeglasses, but I could also use those safety glasses that could come in with your kit. So here they are. And we are working today with hydrogen peroxide, which can be very corrosive. It could degrade your skin. And that is also because of some protein activities and enzymatic activity. That's why you might have a skin reaction. We have vinegar that we are using, acetone that you could find in nail polish. We have some salt, baking soda, a blender, and you have to be careful with the blender. It has very sharp blades, so make sure that you are always not putting your hands into the blender, but be careful about because it might cut you. And then we have some other tubes and a little pipe paper right here, different containers to help us with the reactions, with some tubes containing the various solutions that we might have. And as we are talking about enzyme and talking about food, of course, we could have a little potato here that is going to help us with our experiments. Another thing you could think about for your experiment is an egg. So eggs have in it, like you have egg whites, you have the egg yolk, and in the egg there are of course proteins. And let's say you're frying the egg, your egg white is going to turn white. Of course, why is it turning white? Because the protein in the egg is called albumin is coagulating, and that becomes white. And let's go. We have also a notebook, a lab notebook. And I would encourage you to record everything that you are doing in this experiment, because that is going to help you keep track of what you're doing, why you're doing it, when you're doing it, what did you change, and also what were the results of it. So make sure you write everything from your protocol to your findings in your lab notebook. Now that we know about the materials that we need, we can start the experiment. First of all, I want you to gather everything ready for you to get started. And let's talk about the potato for a little bit. You have your potato here. It's a really nice potato. We cook it, we eat it. And it's very yummy. You can make a lot of things with it. And the thing with the potato, as well as any other foods, starchy food like this, is that they might have some enzymes in it. And these enzymes, we might call them catalase. And they also have a lot of other components to them, which can react with things around us, like oxygens. Let's say we take our potato I have my peeler or a knife if you're using. Be careful with it. You could peel the potato. At first, it looks just white like this, right? But then give it some time, just on in at room temperature with air, and then it becomes kind of brown, and it might turn browner with time, as you can see on here. If you want to accelerate this process, you add salt to it, and salt will make it turn browner faster. So that's one of the effects of salt. But the purpose of this potato today is that we want to extract the catalase, we want to have the juice. So we have it in our blender, cut it up, get it in chunks, put it in the blender, and reduce it to a juice. Now I have my potato juice ready. And as I told you before, potato does react with the air around us. And that's why my juice is a little brown instead of white. And if you're making your potato juice from scratch like me, you might need to have a beaker, a container rather, and cheesecloth or another cloth that you could use to filter out the solid chunks from your juice. So I have my beaker and my cheesecloth, and I'll just pour my potato juice in it. There we go. Make sure you squeeze out all the chunks because it's still a little chunky. And just to ease your process as you're blending using your blender and blending the potato, if sometimes you might need to add a little bit of water to your blender to facilitate the working of everything. So always have your gloves on and make sure you have everything separated. So this is my juice. And now that I have that, I can start the experiment. So first thing you could see, you could try to observe your components. So we have the potato juice here. It's pretty cloudy, like pretty fluid. So now you need to get your pipette and some tubes. Have a nice empty tube here. As you're working with these tubes, make sure you're always labeling the tube so you know exactly what is in what tube and you know what experiment you're doing. So we have our tube. I will label it as potato juice, and I have my pipette. And this pipette, the way it works is that you could squeeze the top and put it in your fluid, and as you release, you would see that your fluid is gonna get into the pipette. The pipette also has some graduation, so you can see exactly of how much you're putting in what. So there we are. I don't need to measure it per se, but I need just a couple drops. And release. And there we are, potato juice in the tube. So I put the juice is in here. And the first thing that I want to do is look at the catalase activity. So one way of doing that is by using hydrogen peroxide. So you will take some hydrogen peroxide that is already in this tube labeled. You just need a couple drops of it. And so you can avoid cross-contamination, I encourage you to use a different pipette. That's why I was saying keep everything separate so you can have a total control of your reaction. So there we go. Measuring some hydrogen peroxide and keeping it there with our older tube. So we can drop a couple drops of hydrogen peroxide. We have, I'm dropping 10 little drops. And as you can see already, it has started bubbling. You see a foam that is rising up. And what I want you to do is let this stay for a little bit until you don't see the bubbling no more. And you're going to measure the height of the foam. There we are, it's still bubbling, and as you can see, the foam is still rising. We're still getting quite a bit of that. And while you're doing all that, make sure you're always recording what you did in the notebook, because in the tube I had about 1 milliliter of potato juice, to which I added 10 drops of the hydrogen peroxide. So I have my table in here already recorded with everything that I've done. And let's say in the process of working here, your hands become dirty. As you can see, my gloves already have some potato starch on it, so I can change the gloves. So one way, if as you're working with chemicals, you don't want your skin to touch anything. So there is a way to remove your gloves without touching it. So this is my hand that has the potato starch on it. Use my hand that is on a glove, put it inside out, and here you're just going from your cuff that didn't touch anything. There you go. You removed everything without touching and replace your gloves. Now, let's say you didn't want to use the potato starch. Maybe you want to use the entire potato by itself. So, as I showed you earlier, I have lamelles of a potato, and you can take this. You could still observe the same process, and put it in one of the nice tubes. Again, you could do a couple things. You could use hydrogen peroxide. You could use acetone. You could use baking vinegar, or even baking soda. It's mixed in water, and just drop your potato or put it in a nice plate. I still have my hydrogen peroxide in here, so I have my potato there, and I could drop another 10 drops of of the of the hydrogen peroxide, and you will see that you are still forming bubbles on the potato, and it's still rising, but not quite as much as with the potato juice. So if you want a very efficient reaction to show, potato juice might be the route to go. But this is a nice example that you could replicate on things other than potato, like grass, and you could also find non-living material like salt, or even like and any other thing that is not food might not have the same reaction, because our potato does have catalysis in them. You could also try to use, instead of hydrogen peroxide, you could try to use acetone instead and observe the same reaction, and find out exactly how the potato is behaving, how the enzymes in it are behaving. And in the hydrogen peroxide, the bubbles are actually oxygen molecules that are broken down from the hydrogen peroxide because of the catalyzing activity from the potato. We have completed 2 experiments. Well, we are still in process. And our third experiment has to do with salt now. So we are still using our potato juice, still evaluating the activity of the catalyzing the potato juice. And you could still get your clean pipette, get some juice, about 1 ml, drop it into your tube that is pre-labeled. And to this, you could add some salt and observe how it reacts differently compared to your hydrogen peroxide. So I have my salt in here. I have my potato juice. Just dropping about a pinch of salt. Close your tube. Mix it up. Make sure that the salt is dissolved. And if you could avoid having bubbles in there, that would be fine. One way of doing that would just be inverting the tube like so. And you could compare it to what you have. So this is our first tube, the potato juice and the hydrogen peroxide. And in our second tube, I have potato juice and salt. And there is really not a real reaction, because all the bubbles that formed in here are bubbles of my doing. They're not coming from any reaction. Even if you compare with just the potato slice, you still have quite a bit of bubbles, smaller bubbles, and in the salt there is nothing. So that means that the enzyme in the potato is not reacting with the salt. So why do you think that is? Could be because the salt is not like is not a living. So because let's say you're breathing. When you're breathing, there is oxygen in the air, and we know that as you're breathing, you're keeping the oxygen and you're releasing the gas. And that is what is happening in this first reaction, where the potato, the catalyze in the potato is breaking down the hydrogen from the oxygen, and the oxygen is released in the form of bubble. That's what you're seeing. Meanwhile, in the salt, there is no such reaction that is happening. The salt is just there, existing with the potato catalyze. So nothing is happening. You can replicate these experiments with grass, with bread, with other enzymatic sources, even other proteins like what you would find in the eggs, especially egg white, because egg whites have albumin, which is a type of protein. You could try and breaking it down. And if you make the egg white react with acetone, you would see something different than if you had made it react with hydrogen peroxide or baking soda. You could try and get your students to make different activity and make different observation. And the goal for them would have been to measure the height of this cone in this first experiment or the second experiment, and also observe what is happening with other different types of agents. So instead of salt, they could be using baking soda. Baking soda has a bit of a different composition when you compare it to salt, because it's not just a salt. It has also a corrosive agent, same as with hydrogen peroxide. So you might see a similar type of reaction. With acetone is another byproduct that could also react with enzyme, as they could denature the enzyme. So if you mix acetone with our potato juice, you would see a different reaction. And one of the things that I told you about earlier in this session was about oxidation. So when you have, like I said earlier, you have your potato and you have air. Your potato reacts with air, and it turns from white to this brownish color that we see in the potato juice. And one way to prevent that from happening is from using ice. So I have some ice in here, and if you put your potato in the ice and keep it there, it's not gonna oxidize. And you can conserve your potato and prevent your enzyme from working. So your enzymes are highly sensitive to temperature changes. That's why in ice it doesn't react, while at room temperature, or like when it's exposed to air, it's oxidizing, and it's becoming, you can say that it's denaturing, because the enzyme is not the same as it originally was. It could be that the enzyme is being chopped up. Let's say you're in a line on a group, you're dancing something, and you're forming a nice circle. As soon as there's an event or some wind that is going to push your line, you might break apart from the person you were holding their hand. And that is the same way with enzymes. It's the same kind of process. The way there is a force that disturbs the whole the line. Now that we completed our first set, we observed how the enzyme in potato called catalase reacts with hydrogen peroxide. We're gonna try to make another experiment to observe how we could alter that reaction to make it faster, perhaps. Now we have our 3 empty tubes labeled. One is gonna be a test tube. We have an acetone control, and then we have a vinegar control. As for the material that we are using our trusty potato juice, which has our catalase, which is the enzyme that we're working with today. Then we have 3 other tubes, which are aliquots of our reagents. So we have some acetone in here, hydrogen peroxide, and vinegar. We are not using the big tube because when you're working with experiments, you have to be worried about contamination. So it's better to take your reagents from a smaller tube, instead of taking it from the source, because if you contaminate something in a small tube, it's better than contaminating the entire batch of reagent. And so what we are going to do, we have our first test tube that we are going to introduce about 10 drops of potato juice. So I'm measuring again with my trusty pipette, as I showed you earlier. 10 drops, or about 1 ml. 10 drops of our potato juice. And we add, again, 10 drops of the potato juice in the control catalase. You can still use the same pipette because you're not putting that pipette in any other thing. And in our last tube, which is our control vinegar, we can add water instead of the enzyme, because this is another one of our control. So here you change pipettes. You have another clean pipette. Get some water. 10 drops again, which is again about 1 ml. Never reintroduce the rest of the fluids in your source because you might contaminate it. So we have our base for all three ingredients. Then we add our vinegar, which is we are testing the effect of vinegar on this on the reaction with hydrogen peroxide. Which one is my vinegar? See, I was fumbling trying to find my vinegar, but my tubes are labeled so I can find it. Make sure you label. So again, we have our vinegar, new clean pipette. Get some. And 10 drops of vinegar, or you could measure exactly. As I said, there is some graduation on the pipette, so you could try and measure exactly 1 ml. And in the second tube it goes, and in the third tube, I add again 10 drops of vinegar. So earlier we measured how much foam was in our tube after our reaction with just hydrogen peroxide and the potato juice. So we are going to be doing the same this time. And I need, again, the hydrogen peroxide to be added to our tubes. And I need 20 drops in the first tube. So that is going to be about 2 mils. And in the second tube, which is our control catalase, I need, again, 20 drops of that. In the third tube that does not have the potato starch, I will add 20 drops of the hydrogen peroxide. And while you're doing all that, make sure you record the quantities in your notebook, because it is important to know what variable you're changing and what you're keeping the same. And in all those tubes, you can realize that the quantities of what I've added to the reaction are the same. In the control catalyze, there's one more thing that we are going to be adding, and that is 10 drops of water. Earlier, we saw how our catalyze in the potato reacts with hydrogen peroxide, and you could observe that through bubbling, which is catalyze breaking down the hydrogen peroxide into hydrogen and oxygen. And the oxygen is what the bubble are coming for. And the way we evaluate that reaction is by measuring how tall is the foam that we observe. Now we are going to do a different experiment where we are going to assess the effect of vinegar on the previous reaction. So we are still going to be using our trusty potato juice with some hydrogen peroxide and vinegar. And this time we are going to add water as a control. So we have 3 tubes, empty tubes, that are going to be our 3 reaction tubes. One that is going to be our test tube, where we are testing actually what does vinegar do to the reaction between the catalase in the potato and the hydrogen peroxide. We have our second tube, which is going to be our control catalase, which is testing what happens if we don't have vinegar in the solution, which is a little bit like what we saw earlier. And then in our third tube, we have another control, which is the water control. And this control only has water, vinegar, and hydrogen peroxide, and no potato juice. So we are not expecting any enzymatic activity in these two. And this is how we could actually assess is vinegar doing anything? Is it reacting with the enzyme in the potato juice, or is it reacting with the hydrogen peroxide? We are going to test all of that. So let's get started really. And here I'm going to add 10 drops of potato juice to our first 2 tubes. And these 10 drops is about 1 ml. And I told you earlier, your pipettes are graduated, so you could measure it exactly how much you need. There we go, 10 ml of that. 1 ml, sorry, not 10. Second, another 1 ml of potato juice. And the third one does not have potato juice, as you can remember. And in the second tube, we are going to add, in the second batch of reagents, we are going to add vinegar. And we need about 10 drops of vinegar into the first tube, which is our test tube. We need again 10 drops of vinegar into our water control, the one that does not have our potato juice. In between, you could pause a little bit to see what it does. And there is no reaction in the two, so it's fine. Then we add our last reagent, which is the hydrogen peroxide. And we need about 20 drops of it, which is 2 ml. So let's start with the control. I have our hydrogen peroxide in here. Nothing happens. We need again 2 ml of hydrogen peroxide for our second catalyzed control. Still the same reaction that we saw earlier. And you see, as I add the hydrogen peroxide, there is some bubbling that starts. And it's bubbling a little bit. It's happening pretty fast. And in our third reaction, again, I have 20 drops or 2 ml of the hydrogen peroxide in our test tube. This tube has vinegar, our potato starch, and now hydrogen peroxide. So there we go. We added our 20 ml. You could wait and observe what happens. There's still some bubbling, but not quite as fast as the first one. So in the tube that only has the potato juice and the hydrogen peroxide, our foam is quite tall. But in this tube that has the vinegar, in addition to the potato starch, potato juice, and the hydrogen peroxide, we don't see anything. In our water control, where we wanted to see if it's if the hydrogen peroxide and the vinegar are reacting, we also don't see anything. So here it can be quite safe to assume that vinegar is preventing the reaction from happening. Now we have done a few experiments testing the activity of an enzyme in potato, which we have been calling catalase, and we have been testing its activity with hydrogen peroxide. And I have in one of these tubes is one of the experiments that we made, and we observed how the catalase reacts with hydrogen peroxide and forms and release oxygen that forms a little foam that grows tall as we go, and it's quite pretty. And we also tested how this enzymatic activity can be slowed down by vinegar in some cases. And you could test it at home, how you could make this enzymatic activity go faster by adding any other reagents like acetone, baking soda, or salt. That is something for you to do at home. And through all of that, we finally know that there are enzymes in the food that we eat, like potato. And thank you for giving us some of your time to listen to our video and go through the experiments. And I hope that you will have a great time replicating these experiments at home or in the classrooms with your students.